All right, this is our last anatomy video. And then we're sort of wrapping up our standard lab sections. After this, we're going to transition more into kind of broad concepts, ecology, symbiosis, reading papers, and doing a little bit of your own research. The research project is not a big deal. I just want you to have the opportunity to read about things that interest you and then share that information with your classmates, and for us to benefit from that sharing. So, not a big time-consuming deal. I know a lot of you have an intense course load, such as the life of a science major. I've been there. So treat it as a project of interest. It is worth 10% of your grade, so don't not do it, but don't stress out about it either if you can help it. Okay, overview. Oh, also, all of these are just rad leaves. Look at this huge water lily. Gigantic. So a little overview. First we'll look at what is a leaf, and then we'll look at leaf anatomy. You did a little bit of this in your lab this week, looking at leaf anatomy. You've already looked at leaf modifications for your last week's lab. So hopefully this will be a little bit of review and a little bit of new stuff. We will have some descriptive terminology, talk about leaf arrangement and parts of the leaf, also in your lab. Look at monocots versus eudicots, and adaptations and modifications. So what is a leaf? The primary function of a leaf is photosynthesis. They are specially adapted to be like solar panels, right? But as we know, leaves have to balance their objectives. I want to be a solar panel and get as much light as possible, but I want to lose as little water from my tissues as I can, depending on my environment. If I live somewhere with constant water, I don't care about that. Often places with constant water are high competition for plants, so they have to have huge leaves to try to get as much sunlight as possible because they're really sunlight limited. Or they have to try to grow on top of each other to get access to that sunlight. Places that live in Southern California, plants that live in Southern California or other places where they're going to have extended dry season, lots of competition for that water, water limited in general, are going to need smaller leaves that have some sort of strategy to prevent water loss. Something you might see for this if you go to the dunes, where you're right by the ocean, there's lots of fog, but it's still a highly drained, fairly sun-intensive environment where plants are still worried about water loss. You'll see a lot of the leaves are pointed perpendicular. If you live more inland, you'll see this in chaparral environments with things like manzanita. You get a lot of this isofacial leaves or perpendicular facing leaves where both faces are only exposed to the sort of rising and setting sun, where it's going through the most atmosphere, it's the cooler parts of the day, that's when you're photosynthesizing, and then at high noon, the hot part of the day, you have less sun exposure, right? So it's a way to limit your water loss, is to have your leaves pointing upright and to have them be smaller. So, leaves, their main objective is generally to photosynthesize. However, plants adapted to stressful environments or with particular environmental pressures might have to use their leaves to do something else, and then some of their other tissues will become leaf-like, usually stem tissue. So, leaves can take many forms and have a variety of functions, as we'll see. You can always tell what is botanically a leaf by where it is positioned on the stem, which is going to be just below the axillary bud. So that position in the node is going to be your way of figuring out, is this a leaf or is this not a leaf? And this only works for angiosperms. Okay, normally I would draw through this cross-section with you. I'm going to try it out just because leaf anatomy is kind of complicated. This is what we're drawing, right? So it's complicated for me to do with my mouse, but I'm going to try. Okay, so on the top, we have a single layer of cells. This is our upper epidermis. I'm just going to label it the UE. In that upper epidermis, depending on where we live, we might have guard cells. We'll probably have some trichomes, some hairs coming out of this structure. But often plants will not have stomata in the upper epidermis because that's where most of the intense sunlight is hitting. I'm not going to write these whole words, but just little identifiers. So we have some trichomes up here, some hairs on the epidermis, and then the UE, the upper epidermis. Just below that is going to be our main photosynthesizing cells. I'll draw these in dark green. They're these column-like cells full of chloroplasts. And I'm just going to kind of more of the same through here. And we'll make some room for a vascular bundle there. Oop. And then more of the same that way. So this layer of cells, these long cells full of chloroplasts, these little stripes are chloroplasts, by the way. This is the palisade mesophyll, and you'll see how to spell this on the next page. But think of this as like palisade, like columns in some beautiful Greek building. So that is the layer that's primarily responsible for the sunlight, the light-dependent part of photosynthesis. Just below that, we'll have another layer of cells with a completely different organization. So these will be loosely packed cells, lots of interstitial spaces, lots of air space in between. They'll still have chloroplasts, they'll still photosynthesize, but a looser arrangement of these cells here. So this is the spongy mesophyll. Think of like a sponge, has lots of air pockets. This is more involved in the gas exchange part of photosynthesis, so the light independent reactions where we're focusing on CO2. So these two make up the mesophyll of the leaf. This is a eudicot leaf, by the way. If we were to look at a monocot leaf, we would just have spongy mesophyll. There's no differentiation between the mesophyll, so we just call it mesophyll, really. Okay, so that is that, and then we have down here another single layer of cells, the lower epidermis, which will usually be where we will find more of the stomata. So I'm drawing these little cashew-shaped guard cells here that are flanking the pores, which are the stomata. This is working out better than I thought it would. So here you have another specialized epidermal cell, guard cell, where it's got a G and an arrow. I don't know if you can tell what those are. And then here would be the hole, the pore, that's the stoma, which looks like 5+. Okay, so we have upper epidermis, lower epidermis, which I'll label LE. The lower epidermis generally has the stomata, but if you're in a place where you have no worries about water loss, you might have equal stomata on both sides. I think it's also more common in monocots to have equal stomata on both sides because they tend to have more upright facing leaves like grasses. Then we'll have, depending on the leaf, you may or may not really have a distinct bundle sheath around the vascular bundle, but I'm going to draw one just because. So this won't always be present or obvious. It is super obvious and distinguishable in corn, which we'll see as our monocot leaf example, because corn is a C4 plant, and C4 plants do all of their Calvin cycle in their bundle sheath cells. So if you remember from C4 photosynthesis, these were the cells that we were talking about of where the Calvin cycle happens and where rubisco is located. So this is the bundle sheath, and that surrounds the vascular bundle. Okay, so for the organization of the xylem and phloem within this vascular bundle, so we're looking at a leaf vein here. Anytime you look at a leaf and you see those veins, this is a cross-section of what we're looking at. So the way that leaves develop is kind of flat against the stem, facing upright, and then they fold outward. which I'm doing this with my hands, but you can't see because I'm drawing. So the leaves kind of face upright with their top facing inward and their bottom facing outward. And if you think about how the xylem is oriented in a stem, xylem is always toward the center, phloem is always toward the outside of the stem, toward the epidermis. So as these leaves develop, the top part of the leaf would be toward the center of the plant, and the bottom part of the leaf would be toward the outside of the plant. And then they'll fold open, and the upper epidermis will be up, and the lower epidermis will be down. But that means that the xylem, which I'll do in red here, we have large vessel elements because we're looking at an angiosperm. Xylem's on top, takes up most of the vascular bundle, and then phloem is down there below, and we would have large cells that were sieve tube elements and small cells that were companion cells. So the blue here is my phloem, and the red is my xylem. Okay, so that's your basic leaf cross section. I'm going to stop the video here, and we can look at the real cross sections and look at those parts.